study the oxidation of fatty acid or fatty acid metabolism. One part is catabolism, fatty acid is oxidized, and one part is anabolism, fatty acids are synthesized. So fatty acid oxidation, the example is 16 carbon palmitic acid, and this 16 carbon palmitic acid having formula CH3 CH2 14 times COOH. Now how this palmitic acid is hydrolyzed in mitochondria to generate huge amount of energy in the form of ATP, and how it is synthesized. So both the points covered in fatty acid metabolism of this palmitic acid with specific example. Large amount of energy generated via ATP hydrolysis. So as compared to the glucose, when there is complete combustion of glucose via glycolysis and TCA cycle, only 38 ATPs are produced. But when single molecule of palmitic acid is hydrolyzed, near about 129 ATPs are produced. So this very large quantity of energy generated via oxidation of the palmitic acid. In 70 kg man, the energy reserved in triacylglycerol is near about 1 lakh kilocalorie. Energy reserved in their muscle protein is near about 25,000 kilocalorie. Energy reserved in glycogen is near about 600 kilocalorie, and energy reserved in glucose is near about 40 kilocalorie. Before starting with the actual lipid oxidation, whatever the triacylglycerol is first of all acted upon by the enzyme lipids to separate free fatty acid with glycerol. So the structure of triacylglycerol CH2OH, CHOH, CH2OH. This is glycerol, but when it combined with the fatty acid, it is like triacylglycerol is like this CH2O C double bond O R1, CHO C double bond O R2, CH2O C double bond O R3. Now this triacylglycerol is hydrolyzed by the enzyme lipase to separate the fatty acid chain. I again repeat, CH2O C double bond O R1, CHO C double bond O R2, CH2O C double bond O R3. These R1, R2, R3 along with carboxyl groups, these are nothing but fatty necessary to separated from the structure of the triacylglycerol to form separate CH2OH, CHOH, CH2OH glycerol along. So consider one of the fatty acid is C double bond O R1. This R1 is nothing but, for example, our palmitic acid. It contains the group CH2 14 times CH3. This is our palmitic acid. So when one single palmitic acid is taken from the structure of triacylglycerol, near about 129 ATPs are produced. Then consider how many ATPs are produced, because 3 fatty acid chains are present in the structure of triacylglycerol. Remaining 2 are there. So 129 into 3, this huge quantity of ATPs are produced when the fatty acids are completely hydrolyzed via beta oxidation of fatty acid, or it is completely catabolized via this degradative pathway or hydrolytic pathway. Up to this clear to all?
Yes.
Up to this clear?
Yes, sir.
Okay. Is it visible?
Yes, sir.
Now clearly visible?
Yes, sir.
Yes. Okay, very
Yes,
good.
sir.
Okay, very good. Now it is beta oxidation of fatty acid. Why it is called beta oxidation? We are going to see one by one. Before actually starting with the topic, we are going to study who discovered this beta oxidation. So before starting with the actual topic, just write down some important points. Discovery of beta oxidation of fatty acid by the scientist Knoop, Knoop. Scientist Knoop discovered this beta oxidation of fatty acid. Then, which type of hormones they are responsible for inducing the hydrolysis of the fatty acid? The hormones which enhances the fatty acid degradation, they are nothing but hormone glucagon, they are epinephrine. So such type of hormones are responsible for some adrenocorticotrophin hormones. They are responsible for oxidation of fatty acid. It is also called beta oxidation because the oxidation or hydrolysis starting from the beta carbon atom. So how this Knoop discovered the beta oxidation of fatty acid? The scientist Knoop fed the dog with phenyl containing derivative of fatty acid. He purposefully fed the dog with phenyl containing derivative of fatty acid. For example, consider if the dog is fed with fatty acid carbon number having carbon number 8, and after digestion of this 8 carbon number fatty acid, when the content of the digestive systems are analyzed, purposefully there is killing of the dog, or they carry out the postmortem and analyze the content of the digestive system, then Loop found that the fatty acids are less in 2 carbon chain. He found 6 carbon structure, then followed by 6 4 carbon structure. So sequentially there is removal of 2 carbon moiety from the structure of fatty acid. Then how it is possible? He checked by feeding the dog with some phenyl derivative of the fatty acid and found that in each round of the oxidation the fatty acid chain is minimized by 2 carbon less. Then he conclude that there is beta carbon present in the structure of fatty acid, and fatty acid is sequentially degraded by removing the 2 carbon structure. So 8 carbon structure is converted to 6, 6 carbon converted to 4, 4 carbon converted to 2. So sequentially 2 carbon structure is removed in the form of acetyl CoA, and then he coined the term beta oxidation of fatty acid by carrying out the experiment on the dog. When we are going to study the different protocol or whatever the steps involved in the beta oxidation of fatty acid, the first point taken into consideration is nothing but activation of fatty acid. Second part is nothing but transport of fatty acid. Third part is nothing but actual oxidation of the fatty acid, and then energetics and so on so. So sequentially we are going to study the beta oxidation of fatty acid. Up till now, any doubt? Any doubt?
No, sir.
Okay. Now see over the presentation. Is it clearly visible to all?
Yes, sir.
Okay. So the first step in the fatty acid oxidation, it is necessary to activate the fatty acid. All you must note down the point that the fatty acid before actual hydrolysis, they are necessary to activate and then transported inside the mitochondria. So as an exam point of view, all you must note down the very important point: beta oxidation of fatty acid takes place in mitochondria. It is very important point. Beta oxidation takes place in mitochondria. But before actual oxidation, it is necessary to convert them into activated form. And activated form of the fatty acid activated form of the fatty acid is called acyl CoA form. So this fatty acid activation, it is a reaction that is catalyzed by an enzyme which is present on the outer membrane of the mitochondria called as acyl CoA synthetase. So the fatty acid which is subjected for the oxidation react with ATP plus coenzyme A to form acyl CoA form. So RC double bond O, this red color portion is nothing but our fatty acid. Consider it is palmitic acid. So R contains CH3 CH2 14 times, C double bond O as it is attached with the S CoA form. So for activation of fatty acid, it is necessary to expenditure ATP molecule, but the ATP is not hydrolyzed into ADP plus PI. The high energy linkage is breakdown into AMP plus PI. So two high energy linkages are taken into consideration. Why it is two, we see in the actual energetic point of view in further energetics of the beta oxidation of fatty acid. But before actual oxidation, it is necessary to activate the fatty acid on the outer surface of the mitochondria, and once it is activated and converted into acyl CoA form, it is now ready to enter inside the mitochondria for further round of degradation or hydrolysis. But the long chain of fatty acid, they are not easily enter inside the mitochondria having carbon number more than 9 or 10. So it is necessary to transfer the fatty acid via the transport mechanism called as carnitine molecule. So with the help of carnitine, the fatty acids are easily enter in the mitochondria, and the carnitine act as a transport vehicle to transfer the activated form of the fatty acid inside the mitochondria. Any doubt up to this? Any doubt? Please ask. Yes. Any doubt?
Sir, please repeat.
When fatty acids are subjected to oxidation or hydrolysis, means for catabolic purposes, they are oxidized in mitochondria. But before actually entering in the mitochondria, it is necessary to activate the fatty acid in the form of coenzyme A attached form called as acyl CoA form. So to convert fatty acid into acyl CoA form, it is necessary to react with the ATP and coenzyme A form to convert the fatty acid into acyl CoA form. Once it is activated, it is easily enter in the mitochondria if having less number of carbon atom in the structure, up to 8 or 9, but more than 10 if the carbon numbers are present in the fatty acid chain. Then for such type of activated fatty acid which is entered in the mitochondria, it is necessary to take help of the one transport mechanism called carnitine, and this carnitine then transfer the activated fatty acid inside the mitochondria. Is it clear?
Yes, sir.
This slide shows the difference between acetyl CoA and acyl CoA. Acetyl CoA is nothing but CH3 C double bond O S CoA, but acyl CoA is nothing but activated form of the fatty acid. So acetyl CoA R is a methyl group, but in acyl CoA form R is a length of fatty acid chain. So can anybody able to tell me number of N present in this given example? CH2 how many times? As we are talking with the example of palmitic acid, how many CH2 are there? CH2 14 times, CH3 1 carbon, so total 15 become. And when we are attached this CH2 14 times CH3 to that R position, by considering the C double bond O carbon, the total carbon becomes 16. Is it clear to all? Is it clear?
Yes, sir. Yes,
I
sir.
again repeat, see on the whiteboard. See here. CH3 CH2 14 times C double bond O S CoA. It is your activated form of the fatty acid which is now enter in the mitochondria for further round of oxidation. So initially it is palmitic acid up to this, but when it is activated it is called palmityl CoA. Now all of you get very clear. Palmitic acid is converted into activated form called as palmityl CoA. Is it clear to all? Is it clear?
Yes, sir.
Yes, sir.
In boys, clear to all? Aditya, Avdut, Shubham, those who are present, please give the reply. Is it clear? But all till this point, all of you not getting the point. What is the actual meaning of alpha carbon and beta carbon? Why from this position cutting is carried out? All of you getting the experiment carried out by the Knoop on the dog? Any doubt regarding the experiment? Is it necessary to repeat?
No,
Yes, sir.
Okay, what did this Knoop scientist? He fed the dog with, for example, 8 carbon fatty acid. But when he analyzed the content of the digestive system of the dog, he found the fatty acid minimized by 2 carbon less, it is 6 carbon. Further, it is converted into 4 carbon. So why in each round of the oxidation, 2 carbon less is there? Why in each round 2 carbon is minimized from the fatty acid chain? This 2 carbon less is minimized because in each round of the oxidation the fatty acid chain is minimized by 2 carbon. In the form of acetyl CoA it is removed. So he gave the name beta oxidation because he do research on this and conclude that from beta carbon position the fatty acids are hydrolyzed sequentially. This is we are going to study one by one. Now clear?
Yes, sir.
Now clear?
Yes,
Yes,
sir.
sir.
Okay. Now again see one by one. All of you got the difference between acetyl CoA and acyl CoA form? Acyl CoA form is nothing but activated form of the fatty acid. So this RC double bond O is nothing but our palmitic acid. Please consider carefully. It is CH3 CH2 14 times C double bond O S CoA is nothing but now palmityl CoA. We are taking of palmitic acid as an example mentioned in your syllabus and it is converted into palmityl CoA. Is it clear? Is it clear now?
Yes, sir.
Yes, sir.
Okay, next. So lipids can be used to make the ATP. This is accomplished through the beta oxidation and it occurs in the mitochondrial matrix of eukaryotes and cytosols of prokaryotes. It is also operated in the small bacteria like Escherichia coli in prokaryotes. I shared one PPT on your group which is different from this PPT. Surprisingly, whatever the data save in our computer, I got this PPT. So actually this live session is going on. I am explaining this PPT whatever the activation reaction we mentioned earlier. Again I repeat, before actually entering in the mitochondria, fatty acid react with the ATP and coenzyme A to produce acyl CoA form, and now this activated form of the fatty acid is ready to transfer in the mitochondria for further round of oxidation. Anyone having doubt regarding this RC double bond O? What is the chain of fatty acid? Carbon number present 16. All of you getting the point? Any doubt regarding this structure?
No, sir.
Okay. Now see how it is transported. Now it is transported via the carnitine shuttle system. How the lipids are moved in the mitochondrial matrix, because fatty acids are transported in the mitochondria for further round of oxidation. Is it clear, image?
Yes, sir.
Okay. Now this RC double bond S CoA is the acyl CoA form activated form of the fatty acid. For time being, the coenzyme A is removed and the acyl form is attached with the carnitine molecule. Then the fatty acid looks like CH3, CH2, CH2. Now this acyl carnitine they transport inside the mitochondrial matrix. Again coenzyme group is attached and it is converted into acyl CoA form, and carnitine again release back and come to the original state again to transfer new fatty acid coming inside the mitochondrial matrix. So for time being, temporarily, the acyl group of the fatty acid is attached with activated form of the fatty acid is attached with that carnitine molecule. So you focus only on the red region. RC double bond O portion is nothing but the activated form of the fatty acid, which is shown in on the slide. It is react with carnitine to form acyl carnitine, and once it is transported, see the reaction number 3, the coenzyme A group again attach with the activated form of the fatty acid as it is previously mentioned in the reaction number 1, and it is transported inside the mitochondrial matrix, and carnitine regenerate back and come to the again original state to the cytoplasm to again enter or again react with incoming new fatty acid. Any doubt regarding this? Any doubt? Yes. Any doubt?
No, sir.
Is it clear to all how activated fatty acid is transported in the mitochondrial matrix from cytoplasm via carnitine? Clear to all?
Yes, sir.
Can we proceed further? What is this? What is this?
Mitochondria.
This is
Mitochondria.
mitochondria. So outer membrane, inner membrane space, inner membrane matrix. So fatty acids are oxidized in the mitochondrial matrix and it is activated on the outer surface of the mitochondria, and once it is activated it is enter inside the mitochondrial matrix for further round of oxidation. I can't proceed complete topic today. We complete this tomorrow, but up to some step, what is the actual meaning of beta carbon atom, how they are hydrolyzed, what are the general reaction, up to this we continue. So it is visible to all what is the actual meaning of beta carbon. Why the term beta is called in beta oxidation refers to the fact that the reaction takes place at beta carbon, which is the carbon that is 2 away from the carbonyl group. Carbonyl group जो आहे सी डबल बॉन्ड ओ त्याच्या शेजारी जो कार्बन आहे तो अल्फा, त्याच्या अलीकडे जो आहे तो बीटा. त्यामुळे बीटा कार्बन पासून हायड्रोलिसिस होणार आहे किंवा कटिंग होणार आहे, म्हणून या पाथवेला बीटा ऑक्सिडेशन असे म्हणतात. आता हे जर एक्झाम्पल पुढे घेतलं तर किती टोटल कार्बन आहेत मोजा. 1 2 3 4, and now one round of oxidation when it is completed it become 2 carbon less. So beta oxidation is a 4-step reaction which is used to break down the fatty acid, and 2 carbons are released per cycle in the form of acetyl CoA. Is it clear? All you get the difference between alpha and beta?
Yes, sir.
One minute. If you are not showing properly, is it clear to all? Clear to all? Hello.
Yes.
Is it clear to all?
Yes, sir.
Yes, sir.
Okay. It is necessary to repeat some points. Any doubt?
No, sir.
Now,
No.
these are the some steps for beta oxidation of fatty acid. These are the some steps. So this is general example, not sorry, general example. It is example of our palmitic acid having C16 carbon atom. Is it visible clearly?
No, sir. It blurs.
Okay. We continue this detail tomorrow, but I again repeat about this alpha beta, how this difference and how from how beta carbon is the chain is hydrolyzed continuously sequentially. So up to the point Knoop experiment on dog, how many calories energy 1 lakh kilocalorie energy stored in triacylglycerol, how lipids are important for hydrolysis, how high large quantity of energy is produced in the form of ATP, how it is important as compared to the glycogen and glucose, how activation is carried out, why it is called beta oxidation, who discovered the Knoop scientists, and once it is activated in the acyl CoA form, how it is transported via carnitine. The beta oxidation takes place in mitochondria. So up to this, all points are clear to all.
Yes, sir.
Okay.
Yes, sir.
Tomorrow maybe on Upadhyay sir's YouTube channel, I am taking live session on this actually reactions of beta oxidation of fatty acid. Is it clear to all?